talking about colon cancer and the effects that modulate cell growth by cell signaling impairment.
Okay, good morning. So today I'm going to talk to you about MYH and colon cancer. MYH is a DNA repair enzyme, and I want to talk about how mutations in MYH might impair important cell signaling processes that might lead to cancer. So human mut-Y homolog is an adenine DNA glycosylase, and 8-oxo-G is one of the most common and stable DNA lesions. As Lauren mentioned earlier, it's caused by reactive oxygen species, and it can be caused by cellular and exogenous sources of DNA damage. And as you know, guanine typically should pair with cytosine. However, when guanine is oxidized to 8-oxoguanine, adenines are preferentially misincorporated opposite 8-oxo-G. So human mut-Y homolog is therefore very important to remove the adenines that have been misincorporated opposite 8-oxo-G. So human mut-Y homolog acts through the long patch base excision repair DNA repair mechanism. And so here, if you have a DNA lesion here, let's say that's the adenine opposite 8-oxo-G, the glycosylase, or HMYH, will remove the adenine and it will leave an A basic site. Then we'll have AP endonuclease 1, which will cleave the DNA backbone. Then we'll have Pol beta, which is a DNA polymerase that will come in and synthesize new nucleotides. As a result, there will be a flap of DNA that exists. Flap endonuclease 1 will then come and cleave off the flap of DNA, and then ligase 1 will come in and seal the nicked DNA. However, it's very important for 8-oxo-G repair to be regulated because you have to consider that you have parental DNA, and you have your parental DNA strand and you have your daughter DNA strand. So here, if we begin with a GC DNA pair, if the guanine is subjected to oxidation, there might be 8-oxo-G that results, and next time during replication, the 8-oxo-G will mispair with adenine. Mut-Y is then very important to remove this misincorporated adenine, and as I just showed in the last slide, long patch base excision repair should occur to reincorporate the proper cytosine. Now, it's very important that this is repaired because if this adenine isn't repaired during the next round of replication, there might be a GC to TA transversion in the genome. And mut-M, which is the bacterial homolog of OG1, will remove the 8-oxo-G and replace it with a guanine. So this is very important because they have found that in colorectal cancer patients, in a certain population of these patients, there are HMYH variants or mutations in HMYH that have led to the cause of colon cancer. So as I already have shown, dysfunctional HMYH may cause GC to TA transversions. There was a study done, a familial study done, where there were biallelic MYH mutations, germline mutations, which led to MYH-associated polyposis. And in these patients, there was a high rate of APC inactivating mutations and KRAS mutations. APC is involved with regulating cellular proliferation in the colon, and KRAS is a proto-oncogene. So the defective MYH was causing further mutations that were causing unwanted cellular proliferation in the colon. And in this familial study, two of the most common HMYH variants that were found were tyrosine 165 cysteine and G382D. And here I just show a colon that is affected with many polyps. And so MYH-associated polyposis is what is most relevant to MYH, and it causes about 1 to 3% of all colon cancers. There's also familial adenomatous polyposis and Lynch syndrome, which are also due to germline mutations. Familial adenomatous polyposis makes up about 1% of all colorectal cancers, and it leads to about greater than 100 adenomas in the colon. And Lynch syndrome, there aren't any as many polyps associated with Lynch syndrome, but in MYH-associated polyposis, it phenotypically looks like familial adenomatous polyposis with less than 100 adenomas in the colon. So these are all mutations within the HMYH gene that have been found in MAP patients. The mutations in white have all been shown to contribute to the pathogenicity of colon cancer. However, there are many other mutations here that have been found in MAP patients that haven't been characterized biochemically. So a large part of my research, I would like to look at some more of these mutations and determine their biochemical activities, their enzymatic activities, and whether they contribute to pathogenicity and may lead to colon cancer. So my hypotheses include that MYH is highly coordinated with multiple cellular pathways to properly regulate ADOX-G repair, and that if there's disruption of protein-DNA interactions or protein-protein interactions between MYH and cell signaling pathways, that adenine lesion repair will be impaired. And finally, disruption of protein-DNA and protein-protein interactions will inhibit or dysregulate redundant or overlapping repair processes. So I'll share with you some of my preliminary results. So here, this is full-length HMYH, and here I show that there are 5 very important protein-protein binding sites. At the N-terminus, replication protein A interacts with HMYH, and at the C-terminus, proliferating cellular nuclear antigen also interacts with HMYH. Both of these proteins are involved with DNA replication. Here, MUT-S alpha, which is another DNA repair enzyme involved in a different pathway, interacts with HMYH, and AP endonuclease, which is involved with the DNA repair process that I showed earlier, interacts here, and HUS1, which is involved with the 911 cell cycle checkpoint process, interacts with HMYH here. In my lab, we've worked with a smaller fragment of HMYH, which I'll refer to as HMYH delta C5, and it maintains 3 important protein-protein interactions here. So we have been successful in crystallizing that HMYH delta C5 fragment, and we were able to collect X-ray crystallography data at UMB. And furthermore, I was able to take the crystals to the Brookhaven National Laboratory in New York and to run further experiments so that we can hopefully determine the structure of this MYH delta C5 fragment. Now, importantly, MYH has an iron-sulfur cluster, which enabled us to complete a single wavelength anomalous diffraction study to allow us to solve the phase problem in crystallography. So we were able to collect data, and this is an example of some of the reflection data that we collected. And we collected data to 2.3 angstrom resolutions. And so basically, this is just showing that we're on our way to collecting data that will help us solve the structure of this MYH delta C5 fragment. We've already begun to work on the model here, and here's some electron density, and we show the iron-sulfur cluster that's coordinated with MYH delta C5 here. After that, we also wanted to show that MYH delta C5 is a catalytically relevant and active fragment. So we completed a glycosylase assay to show that the MYH delta C5 fragment can work on DNA substrates to remove adenines opposite ADOX-G. Here, we reacted the DNA substrate with the MYH protein at 37 degrees Celsius, and we were able to run the reaction samples on a denaturing 14% polyacrylamide gel. And if this reaction was successful, we would have a 10 mer product that would show up further down on the gel. So here we are able to show that MYH delta C5 was able to generate this 10-mer product as we increased concentration of the protein. Mouse MYH, I show here to compare the activities between MYH delta C5 and mouse MYH. And importantly, I just want to mention that we haven't been able to purify large amounts of the intact human MYH, and that's why we have been working with this fragment instead. In addition, we wanted to do some DNA binding studies. And so what we showed here, we have another DNA substrate, and we interacted with MYH. And when MYH binds to the DNA substrate, because of the iron-sulfur cluster in MYH, it's able to quench fluorescence in the DNA substrate. And so we were able to determine some KDs and examine the DNA binding properties of MYH delta C5. So far, we have crystals of MYH delta C5, which have diffracted to 2.3 angstroms, and we're working on constructing the model. And we have preliminary glycosylase assays and DNA binding studies, which have been successful to show that MYH delta C5 is catalytically active and binds DNA. And we are currently developing protein purification and binding affinity assays to study the key protein-protein interactions that I was talking about earlier. And some of our future directions include additional crystallography endeavors. We'd like to examine MYH delta C5 bound to DNA, and also we want to characterize more of these MYH-associated polyposis MYH variants. And I'd like to thank Dr. Toth, who is my principal investigator. Dr. Lu Chang is the PI who is in the lab that we coordinate with. Dr. Golishi is the postdoc that I've been working under. Tamika Patterson is the lab tech. Jade is another graduate student in the lab. Dr. Gerald Wilson and his lab members have been helpful in providing me suggestions as I've been working my research. And of course, I'd like to thank the Meyerhoff program. Thank you. Well, those were all due to germline mutations. So in the other percentages of colon cancers, a lot of times like APC, for example, which regulates cellular proliferation in the colon and was attributed to causing the FAP, that's also mutated in other colon cancers, but they're somatic mutations. So there's been other things in the environment that may have caused these mutations. So these are just going to give us some insights about colon cancer in general. But,
So KRAS is also involved in other cancers as well, and I'm not sure if APC is or not. But I think it is. So is MYH, are there mutations in other types of cancers besides colon cancer?
They haven't found any, and I was wondering about that, and I'm not sure if it has anything to do with the oxidizing environment of the colon. But I don't know, like the lungs are also, subjected to a lot of oxidative stress, so I don't know why that wouldn't cause perhaps lung cancer there, but MYH associated colon cancers have only been found in the colon. So.
Any other questions? No. You mentioned that you were unable to purify the human sample of HMYH. Do you know why?
We don't know why, but we are trying different techniques. We've been trying to grow it up in bacteria, but we're going to try to grow it up in yeast as well and see if there's different cellular machinery there that might enable us to get higher amounts of the human protein. But we don't know exactly, so that's why I've been working with the fragment. But other species like mouse MYH, they've been able to purify the full-length protein. So.
So I'd like to thank, yes, sorry.